Bismillah Rahman Rahim. We start the part two of the question paper which I have just discussed in one video. And this is the question 4, 5, and 6 which follows that video. And this is the Biology 9700 syllabus, the present 2022 syllabus, which I'm going to be talking about in this paper, which are the three questions. Now starting with question number 4. A part, figure 4.1 is a drawing made by a student of a plant cell showing one of the stages of mitosis. Describe the role of the spindle during the stage of mitosis, and this is for 2 marks. Then the chromosomes are composed of DNA. Describe the structure of a monomer of a DNA molecule. So a monomer of a DNA molecule. Now if you know that, I mean, only then you can do this, otherwise you're going to be really having problems in understanding the question. Now as you go on to read the rest of the question, telomeres are located at the ends of each chromosome. Telomeres progressively shorten each time. So the ends of the chromosomes, now we've got the egglets which we talk of, the shoelaces. These are the telomeres. And telomeres progressively shorten. With the time they will become lesser and lesser. So what we have to understand is that as it says telomeres progressively shorten, each time a healthy human cell divides. A healthy human cell divides until the cell is no longer able to divide. So read very carefully. Some cells have an enzyme called telomerase that prevents the telomeres shortening. So it will not shorten and it will remain, because the telomerase is working, so the telomeres will remain, they won't shorten during the cell cycle. Suggest why telomerase activity is important in stem cells, 2 marks. And suggest why tumor cells produce high concentrations of telomerase. Scientists have reached the possibility of treating cancer by inhibiting the action of telomerase. One method that has been studied involves inserting small single-stranded DNA into cancer cells. These sections of DNA have a base sequence that is complementary to a section of the mRNA transcribed from the gene coding for telomerase. Explain how inserting small single-stranded DNA could prevent translation of the mRNA to produce telomerase. Okay, we come back to this a little later. Let's just quickly revise mitosis. So in mitosis, the first phase is prophase, and as I tell you always, is prominent. Prominent means that the chromosomes have condensed, shortened, and condensed, so they become more prominent. In metaphase, I always say this is the middle. So it's actually, the word middle is not to be used, but we use the word equator for it. But just to remember it, metaphase, middle. Then anaphase, they move apart. A for anaphase, A for apart. And then telophase, where are 2 nucleuses. And it's still one cell, but there are 2 nucleuses in it. So please remember, there's a very interesting MCQ on this in one of the past papers. So just a quick revision of prophase, prominent, metaphase, middle, anaphase, apart, and telophase. One cell having 2 nucleuses, because as yet, now this whole thing is mitosis, and then of course cytokinesis is not part of mitosis. Then the machinery of mitosis, the spindle in animal cells, the spindle fibers are anchored at either end to a centrosome, and this is the centrosome, which we've got to the centriole, and the centrosome is the area around it. And each centrosome is composed of a pair of centrioles. So this is that pair of centrioles that you see here. There are 2 like this structures. There are 2 rod-shaped structures, and these are called the centrioles. Then plants lack the centrosomes, and their spindle formation is poorly understood. In all the cells, the opposite ends of the cells are referred to as the poles. So you don't say ends, you say poles, and that is the important word that we use, that the chromatids separate and then move to the poles of the cell. Then coming to another important point, centrioles, they are animal cells only. Centrioles are made of 9 triplets of microtubules. So this is one triplet, this is the second triplet, this is the third triplet, fourth, fifth, sixth, seventh, eighth, and ninth. So 9 triplets, and they're part of the cytoskeleton and help support the cell. Spindle fibers are elongations of the centriole and form the mitotic spindle. Now, as you look at this question 4, and that's a drawing made by a student of the plant cell. Now you can see now they've separated, so this is a part, so it's anaphase. But they are not asking you this question. They're not asking you about anaphase. Well, they're saying describe the role of the spindle during the state of mitosis. So spindle fibers attach to the centromere, and this thing is the centromere. This structure here is the centromere. Centromere is the part where it's a non-coding region, where the two sister chromatids are attached. So centromeres and the fibers contract. Now the spindle fibers contract. Contract means they shorten. So they're going to shorten somehow because you see they're going to be pulling. They're going to be attached here. They're going to be attached here, and they're going to be pulling these to the pole. So they're going to be pulling, and then we're going to have some. Of course, they're not going to be any centromeres here because this is a plant cell. So they are no centrioles, sorry. So spindle fibers attached to the centromere, fibers contract or shorten or retract. Please do not use any other word. I know I've always told you not to use the word contract. It's only muscles contract. But here it was allowed. Spindle fibers do contract. The word contract has been used. And this will pull the daughter chromosomes. You see, it's either you say the sister chromatids or you say pull the daughter chromosomes to the opposite poles. That's the word that you don't say opposite ends. The word poles has to be used, and that's the correct biological English. Then we come on to the next part. I read this question before also. Describe the structure of the monomer of a DNA molecule. Now, I would always do is I would just draw a diagram on it. I would not be bothered about, writing a lot of stuff. I would, of course, have done this 1, 2, 3, 4, 5. And to one could be attached any base. You could say adenine, you could say thymine, you could say cytosine or guanine. I'm not saying you have to say this. And to this would be attached a phosphate. So I would write here phosphate, and that would explain it. So as you can see in the mark scheme also, it's very clear. It says deoxyribose and phosphate and a base. So I would also give a little explanation here, and I would say nitrogenous base, and I would make sure that I got my 2 marks so that I didn't make any mistake. I would just write a lot to be sure that I would fit in the mark scheme and get all the marks for it. So deoxyribose and phosphate and base, and one from the, you can give it from the diagram, the phosphate group and the base attached to the deoxyribose, and one correctly named base, A, T, C, and G. So you could have said adenine, you could have said thymine. Please get the spelling right. Thymine is thymine, and cytosine is Cytosine, and guanine is Guanine. So guanine. So these are the 3 names. Please get the spellings right. Coming to this part of the question, suggest why telomerase activity is important in stem cells. Now, if you remember, stem cells is part of the a very previous chapter in which the stem cells are the ones which are responsible for all the development of the different organs of the body and the differentiation. So basically telomerase activity, you see, because it says the cell is no longer able to divide. This was the point that you had to remember. So this is telomerase activity, if it allows, will allows DNA replication to take place. So DNA replication is very essential because DNA replication has to take place only then of course S phase of interphase, and then mitosis can occur. So allows DNA replication to take place, then stem cells keep on dividing. The ability of stem cells to divide is unimaginable. So stem cells self-renew, or they keep dividing. These are the words that the mark schemes are making you use. So please be very aware of what the words are. Keep dividing, or you can say self-renew. Then stem cells differentiate into different organs, as I said earlier. Stem cells differentiate. So differentiate again is a word, differentiate into different tissues and organs of the body, into specialized cells. Differentiates into specialized cells. Like, the cells will develop into the organs of the body, arteries, veins, heart. So these are all the different parts of the body which they're going to develop. And of course, here they're talking of the plant cell, but otherwise they're talking generally about the different cells. So allows DNA replication, stem cells keep dividing, stem cells differentiate into specialized cells. Then you could have also said that mitosis produces cells for cell replacement. You see, because it said why telomerase activity is important, because it said telomerase progressively shortens. So you had to go back to why telomerase was important. So mitosis produces cells for cell replacement. This is, of course, all the role of mitosis, which is part of the syllabus. So to have cells for cell replacement, and basically they're asking you the role of mitosis. So cell replacement or tissue repair, we can say that. And of course, this also prevents the loss of genes, because you see, if the telomeres shorten, then we're going to loss of genetic information is going to take place. Previously, they were this long, the telomeres, and now they have become shorter. So this is loss of genetic material, loss of genes. So prevents the loss of genes, right? Two of the question was very relevant. Suggests why tumor cells produce high concentrations of telomerase. So at the first, the tumor word comes into your mind, and you think, "Oh my God, what is a tumor?" And you just write whatever you know about tumors, and you get the mark for it. Allows uncontrolled cell division. That's a word I use a lot when I'm teaching you this. Allows uncontrolled cell division. Allows uncontrolled cell division. And that, of course, results in a tumor. And this is actually shortening, allowing shorter cell cycles. Very interesting question. I like this a lot. Scientists have researched the possibility of treating cancers by inhibiting the action of telomerase. So we're going to stop the action of telomerase. Now, telomerase, remember, is a protein, so it must be coded. There must be a gene for telomerase. So one method that has been studied involves inserting small single-stranded DNA into cancer cells. Okay, now look at what I'm going to do. I'm going to explain this to you. This is a cancer cell. This is the nucleus, right? So there must be a gene for telomerase. So that unzips, the mRNA copies it, and the mRNA leaves the nucleus. And the mRNA is to go and attach on the ribosome, right? And the enzyme telomerase will be made. This is the enzyme telomerase will be made. But we don't want it to be made. Scientists have researched the possibility of treating cancer by inhibiting the action of telomerase. One method has been involves inserting small single-stranded DNA into cancer cells. Okay, so single-stranded DNA. So what do I have here? I have AAATTT or anything AATTTCCC or something like that, or AACCC or AAGGG. Now what is this? This is a ssDNA. What do I mean by ssDNA? It doesn't mean super science. It means single-stranded DNA. So I have ssDNA into the cancer cells, and I'm going to throw this. I'm going to see that somehow I get this into a cancer cell, because this was a cancer cell which I just drew. Now, when I throw this into the cancer cell, what is going to happen? These sections of DNA have a base sequence that is complementary to a section of the mRNA. So this is complementary to this mRNA which I just showed you. So that means the mRNA, what will be complementary to this? U, A, G. Now, when this mRNA comes out, now look how it's going to be very clearly understood. This mRNA, when it comes out, it is this mRNA. Now, what is going to happen is these single-stranded DNAs are going to attach to it. So there's going to be attached to it. So there is going to be no translation. Well, that was a very interesting question, because it really checked how good you are in comprehending, how good you have, how much have you understood biology. So these sections of DNA have a base sequence that is complementary to a section of the mRNA transcribed from the gene coding for telomerase. Explain how inserting small single-strand DNA could prevent translation of the mRNA to produce telomerase. Very simple. DNA binds to the complementary section of the mRNA. mRNA cannot bind to the ribosome. Ribosome cannot move along the mRNA. No site for tRNA to bind. tRNA anticodons cannot form hydrogen bonds with the mRNA codons. So you have to understand is that there's going to be when this whole thing happens, the mRNA binds with the single-stranded DNA which we have inserted. This is the single-stranded DNA which we have inserted into the cancer cell. So there'll be no translation. So telomerase enzyme will not be made. No telomerase. So maybe we can solve the problem of cancers and we can treat cancers like this. Right? So DNA binds to the complementary section of mRNA. This is here what I just drew it here. mRNA cannot bind to ribosome. So now this is blocking it. So the mRNA cannot bind to the ribosome. Ribosome cannot move along the mRNA. So my ribosome cannot read that. What is the consequence? No site for, you have to remember, is on the mRNA you have the codons. This is one codon. And to this attaches anticodons. So the no sites for tRNA to bind to the mRNA में codon है. So the codon is on the mRNA. tRNA anticodons cannot form hydrogen bonds with the mRNA codons. mRNA is the codon, but then the tRNA has to bind to it. So very, very interesting question, and I hope all of you have understood it and it's clear to you now. Student investigated question 5, the effects of an air movement on the rate of transpiration by using a pottometer. Define the term transpiration, loss of water vapor from the leaves. The pottometer used by the student is shown in figure 5.1. The student recorded the distance. The air bubble of the apparatus had moved after 10 minutes with no air movement. The rate of uptake was used as a measure of the rate of transpiration. The apparatus was then used using the reservoir and the experiment was repeated at a further 2 times. All other variations were standardized. Table 5.1 shows the results recorded by the student. So experiment 1, distance moved 12.5, 12.0, it's in millimeters. Now this is actually not in the syllabus anymore, but I'm just talking about it for a little point and I'll explain to you why. This is now no longer pottometer and this whole thing is now not in the syllabus anymore. Use table 5.1 to calculate the mean rate of movement of the bubble, that was 1.2, and this is the working of it. You added up 36 divided by 312 divided by 10 because we had 10 minutes here. And here we are asking not for 10 minutes, we are asking for millimeters per minute. So there was just a little bit of maths in that, and I hope everybody got this right. Now the student carried out another experiment using the same plant. In this experiment, a fan was used to blow air across. All other variables were standardized. The result showed the bubble moved further in 10 minutes. The student concluded that air movement increases. Now, this is actually not in the syllabus anymore. So, this cannot come in your new syllabus for 2022 and how air movement increases the rate of transpiration. So, I'm just going to skip that. I'm not going to talk about that a lot. Explain how water moves up through a xylem vessel in the stem of the plant. Now, this is in the syllabus. That part was not, but I just talked a little bit about the maths part of it. Explain how water moves up through a xylem vessel in the stem of the plant in the pottometer shown in figure 5.1. Now, I'll try my best to explain these points to you. The movement of water up the xylem vessel is very much in the syllabus still, and so the movement is by cohesion tension. Now, what is cohesion? Cohesion is the attraction between the water molecules, and that is because of the hydrogen bonds between the water molecules. So, cohesion, I said slash hydrogen bonds between the water molecules. These are called the cohesive forces. Then water moves as an unbroken column, like a continuous column of water. Then the transpiration pull, the fact that evaporation is taking place from the leaves, so that is creating a sort of a suction force. So transpiration pull, then adhesion of water molecules to the cellulose. Cellulose is in the wall of the xylem. There's only the cell wall left. Everything else is finished. The cell membrane is gone, the organelles are gone, the cell contents die. That's what we say about the xylem. So adhesion of water molecules to the cellulose has a hydrophilic property. so water loving property. And the evaporation lowers the water potential. The word I have used water, I've used the symbol for it, lowers the water potential at the top of the xylem. So the evaporation lowers the water potential at the top of the xylem vessel, and that of course makes the water move down a water potential gradient from a region of high water potential to a region of low water potential. So there were only 4 marks, but of course the marking points are many. But please remember, when you write more, then you stand a very good chance of getting the 4 out of 4. If you've written 6 points and maybe one of them is wrong or two of them are wrong, but you've written more than what you actually wanted, that is going to be a win-win situation for you. So remember that and keep that in mind. Now the last question, question 6, figure 6.1 is a diagram of sections to the heart showing 2 stages of the cardiac cycle, A and B, stage A and stage B. Now it says name stage B in figure 6.1. State one piece of evidence from the diagram that supports your answer. So of course it's atrial contraction. Why? Because you see the blood is flowing, the atrioventricular valves are open, and the blood is flowing from the atria to the ventricles. And look at I've shown you in the green and the yellow. So you can see in the green how the atria is slightly bigger here. And here in this one, the atria is slightly smaller here. So the reason is it's atrial systole is atrial contraction. Atrial systole means atrial contraction. So, I mean, systole word is for contraction. If I say ventricular systole, I mean ventricular contraction. And then, of course, it says why, and you have to give me some proof. So the volume of the atria is smaller, and the AV valves, the atrioventricular valves, are open. So blood is flowing. So blood is flowing from the atria to the ventricles, and this is what you're seeing here and here on both sides. So the atrioventricular valves are open, or you could have said the bicuspid and tricuspid valves are open. You could have used those words, or you could have said atrioventricular valves. Of course, you can't write this abbreviation. I would rather you prefer to write the atrioventricular, the whole story, so that you get the mark for this, because AV is not an allowed abbreviation. And the blood is flowing from the atria to the ventricles. So you can see the blood, you can see the arrow blood flowing from atria to ventricles. So you can see here I've completed the mark scheme. Of course, there were only 2 marks, so you have to give me atrial systole would get you a mark, and any one of the explanation would get you a mark. So be very, very careful when you're writing your answers, so that you know it. But if you didn't write it, I'm sorry, I'm not going to give you any marks. Draw a label line on figure 6.1 to identify one semilunar valve. Label the valve S. So semilunar valves now, of course, is what we have to understand. The semilunar valves are very easy to identify. See, I've marked them in red, the semilunar valves. This one, of course, here in stage B, you can see this one is coming out the right side, so that would be the pulmonary artery. This one, of course, is the aorta coming out. So you can see this one is the one here coming out here, from here to here. Of course, I've just crossed it on that. So this would be the aortic valve, and this would be the pulmonary artery valve. So the aortic valve comes out of the left ventricle. This is the left ventricle, and this one is coming out of the right ventricle. So that would be the pulmonary artery. Then, as we go along in the question, describe the role of the semilunar valves in the cardiac cycle. Last part of the question, that was 5 marks, was an easy question as far as I'm concerned. So simple answers: prevent blood flowing backwards from the arteries into the ventricles. So if the semilunar valves close, then of course the blood is not going to come back from the pulmonary artery into the right ventricle, or from the aorta into the left ventricle. Or you could have said, there's an or here, ensure blood flows in one direction and from the ventricles to the arteries. Yes, when the semilunar valve will open, then the right ventricle blood will flow into the pulmonary artery, and from the left ventricle it will flow into the aorta. So either of these answers was correct. I'm just going to revise a few of the micrographs with you, and that will be all for this paper, and that completes this paper tonight. And please do give your comments, and all the very best. Just a quick look at some micrographs, and you can see this is the Golgi. And you've got to understand that how the Golgi is, the shape of the Golgi, what it looks like, and how. So this is the Golgi, then this is mitochondrion. You need to do a little research on this. Of course, you can see another animal cell electron micrograph, and you can see this is the nucleus, which I always say gives you this mottled appearance. Here, there's the nucleus here, and this, of course, is the nucleolus. And of course, this is another diagram which is of course labeled, and I want you to go through it and see it. And of course, The more micrographs that you see, the easier it will be then for you to identify them in the MCQs and the paper too. So just a few of them which I have shown to you here, and please do revise this very thoroughly. And then, of course, I think all the MCQs and other on the micrographs will become easier. So thank you once again, and I hope this is helpful.